We now come to tonight's presentation, which I'm very pleased to tell you is given by Professor John Rasko, AO, Head of the Department of Cell and Molecular Therapies at Royal Prince Alfred Hospital. So, and you do remember, if you have questions that arise while it is on, it's good to get in with, put your questions on the Q&A, and we will have a Q&A session at the end. So John Rasko is an Australian pioneer in the application of adult stem cells and genetic therapy. Since 1999, he's director of the Department of Cell and Molecular Therapies at Royal Prince Alfred Hospital, and also at the Gene and Stem Cell Therapy Program at Sydney University. He's currently president of the International Society for Cell and Gene Therapy. So I think you can believe that he is a real expert. His full CV is written out on the agenda papers and in the advertisement. He's a founding fellow. I will just be brief. He's a founding fellow of the Australian Academy of Health and Medical Sciences, and in 2018 he was the ABC's Boyer Lecturer, and he was made an Officer of the Order of Australia in 2012 for service to the biomedical research in the field of gene and cell therapy as a clinician, author, administrator, and philanthropist. What a record. I'm so pleased to be able to present to you tonight Professor John Rasko to speak about the dawn of molecular medicine, gene therapy, past, present, and future. Thank you.
Thank you so very much, Ian. Thank you, everybody, and I'm very humbled by your kind words, Ian, and very pleased when the invitation from Judith and Christina came by to deliver this address to this august society, which I hold in great esteem. So many friends and members I've seen, and fellows, I should say, with names I have seen already on the list. I'm absolutely humbled to join you tonight, and also to acknowledge, along with Ian, the traditional owners of the land, the Indigenous peoples, past, present and future, from, in my case, where I am, the Gadigal people of the Great Aora Nation, and I acknowledge those custodians of our land. Tonight, I have set myself the task of introducing a biomedical topic to this diverse group. And so I know there are people amongst the audience, I'm looking at you, Susan, who know at least as much as I do on this topic, but also there are individuals who have diverse interests, but all of us are joined together by empiricism, by a commitment to science, and I do acknowledge that that is the underpinnings of the Royal Society, both of New South Wales and indeed the Royal Society itself. So I've set myself the task of talking about gene therapy and its partner cell therapy and giving you somewhat of an overview. But before I do, I'll make just a couple of disclosures, simply that I do receive some honorarious speakers' fees and participate in clinical trials about which I'll be speaking tonight from a series of different commercial entities listed here, and there are probably others that I've overlooked. This is not exhaustive. I serve as a director of pathology. I'm a hematopathologist, a hematologist clinically at a company called Genea that does in vitro fertilisation. And I've consulted in the past for a company called Raresight with stocks in lieu. Importantly, I also have been for over a decade the chair of the Gene Technology Technical Advisory Committee of the Office of the Gene Technology Regulator, which is Australia's regulatory agency for all genetically modified organisms, be they plant or animal. And in that role, I should emphasise I do not speak, therefore, on behalf of the Australian government. I've set myself this topic to introduce gene therapy, and I've divided the talk into 4 parts, as you can see here. I want to give you a reality check to start with, to position where gene and cell therapy is, and of course give you some definitions. Then I want to give you some examples to make it real, to show you where we really are and what has been achieved over the last few decades. and finally close with just a brief acknowledgement of the responsibilities that clinical innovation like these bring. And so I start at, I think, perhaps an unusual place, which is a quote from the Food and Drug Administration of the United States of America, past Commissioner Scott Gottlieb, who entered the revolving door of the Trump administration and exited within about a year, but did a very good job while he was there. And early last year, he announced that by 2025, the number of approvals for clinical cell and gene therapies will increase by up to 900%. Now, that was seen as a bid for further funding for the FDA, but it was also a realistic test of where the field currently thinks it is going. And I hope to convince you tonight that we are on track in terms of accruing new clinical therapeutics that will be approved by rigorous regulatory agencies, including the FDA and indeed the equivalent in the EU, the equivalent in Australia and other Southeast Asian nations, to deliver curative therapies for a number of genetic and acquired diseases. But I get ahead of myself. Let me start with the question that hopefully is burning in all of your minds, which is, what are cell and gene therapies? And so to start, we have to start with the cell, which is one of the wonders of the planet and indeed of all life, the foundational unit of all life, capable of binary division and thereby giving rise to every cell in not only a human body, but every other body that we know of. That miracle of nature gives rise to both somatic cells in the body that provide for all of the different tissues in our bodies, but also it gives rise to germ cells and other forms of stem-like cells that can propagate themselves and also propagate our species. Within that cell that was identified in the 19th century by luminaries such as Rudolf Virchow and others, we now know that there is genetic information that is propagated from cell to cell, and that, of course, is revealed when we look very closely into the nucleus of the cell as the DNA, identified in 1952, 53 by Watson and Crick as deoxyribose nucleic acid. But of course, prior to that, we knew that that was the genetic material, and that is the material that gives rise to inheritance. And it is the same material, of course, that gives rise to potential errors, and those errors can lead to both somatic diseases, in other words, diseases arising in the body, such as cancers that may be environmentally induced, or it can lead to inherited disorders, genetic inheritance by mechanisms that have been well characterized that lead to the propagation of disease. So that's what a cell is and the genetic material in a cell. What we want to do in cell and gene therapies is use this living material, a new mode of medicine, a living propagating material to provide potential cures or therapies. And in order to do that, we want to try and either correct or add to the genetic material in a cell using various different technologies that are available to us and have been developed over time. And the idea here is simply, if you have a genetic abnormality, whether it be acquired or inherited, you may be able to inject a form of DNA or DNA itself into the cell by various technologies, in particular mechanisms that based on viruses, that would lead to a permanent genetic change of that cell, and if it is a stem cell, the propagation of that cell. I would emphasize at this point that it is not legal in most jurisdictions worldwide, and including in Australia, to genetically modify germ cells. In other words, those cells which will give rise to our progeny. So what I want to convince you of for the rest of the talk is that gene therapy is a disruptive technology because it is a living technology. Gene therapy and cell therapies go hand in glove, as I've just demonstrated in the animation, because the genetic material of all cells is indeed what is propagated as the informational component. And we now have harnessed the technology to be able to modify that. The idea is compellingly simple and straightforward. Namely, if you accept that many diseases are caused by genetic abnormalities, then the idea is let us alter or correct that abnormality and see if we can effect a cure. In the remaining part of the talk, at least in the middle, I want to convince you that we are now achieving clinically meaningful results. But before I do, I want to make a distinction between two different types of gene therapy. We have the opportunity to be able to genetically modify cells either inside the body directly, that's called in vivo gene therapy, indicated on the left side of the slide, or we can remove cells from the body and genetically modify them outside of the body, and then either store them indefinitely in liquid nitrogen or replace them whilst they are still alive into the person's body and affect potential therapeutic benefits if we are able. So those two forms of gene modification ultimately reside in cells, but they can be affected in vivo, where we can inject into tumours, we can inject intravascularly, and the virus then homes to various target organs, or we can inject into other tissues, living tissues like muscle, or we can take the cells out of the body. A couple of years ago, colleagues and I in the International Society for Cell and Gene Therapy set out to define what cell, tissue, and gene products worldwide had achieved marketing authorization. And this is basically an amplification of the information I gave you in the quote from the FDA, showing that over the years since 2001, there has been a steady increment in the annual approval of cell, tissue, and gene products worldwide. Some of you may be surprised to even see that number 44. I might say to you that it's quite low, but others would say that they are shocked to hear that there are any such approvals. And I would emphasize the fact that a fraction of those are due to bone marrow transplantation, because in the United States of America, at least, so-called cord blood transplantation is approved as a cell, tissue, and gene product. Nonetheless, this is evidence that there is an accrual of increasing numbers of approvals that pass through an extraordinarily rigorous regulatory pathway in all of the different jurisdictions worldwide. Going further, we can look around the world and ask the question, where are those approvals occurring? I've mentioned the United States of America, where the number is 16, as you can see there. But look in our region of the world to South Korea, which has got a very receptive national program in approving cell gene therapy products worldwide. And you can see it is a dominant player, especially when you reflect on the fact that it does not approve bone marrow transplantation as one of its categories. In the EU, there are also representations, and as you can see, we have at least one or now a few accrued in Australia. I would emphasize these are data that are a couple of years old. I think it gets even more interesting when we ask the question, well, what are those approvals? And so you can see here on the left pie chart that there are basically a distribution between half of these cell tissue and gene therapy types are in cells from one's own body, and the other half cells from another person's body, a so-called allogeneic source. In the middle pie chart, it's not a surprise to see that the majority of these 44 approvals are cell-based therapeutics, and I'll elaborate on that later. But what I want to emphasize in this slide and one just to come is that there is a great diversity in the disease types for which these approvals have been granted. In other words, it's not just one disease type or one clinical specialty, my favorite, of course, being hematological, but it is a diversity in eye diseases, degenerative neurological diseases, immunological diseases like boy in the bubble disease and severe combined immunodeficiency, and so on and so forth. A diversity of clinical disease types and clinical specialties which have mastered this technology and received approval. And here are just a couple of the names of some of the approvals of cell and gene therapy products in the United States of America, for example. You can see Luxturna for blindness was approved a few years ago. When I was asked some years ago about where I thought the future of gene therapy was headed, and they said, well, go crazy, John. Tell us what you think is impossible to treat in gene therapy. I would never have guessed back even perhaps 10 years ago that we would be in a position to cure an inherited form of blindness, but now that is the case with retinitis pigmentosa. We are able to cure that inherited disease, which would otherwise cause legal blindness in children and teenagers who are born with this rather devastating inherited abnormality. The last two on the list are ones that I'll touch on later. Kymriah and Yescarta are their commercial names, and then of course the more unpronounceable professional names, Tisagen Leclucel and Axicaptogene Siloleucocel, which are both approved in Australia. Yescarta emerging as an approval, and Kymriah now approved in Australia for the treatment of acute lymphoblastic leukemia and forms of lymphoma. When we focus on the gene therapy ecosystem, many of you won't be able to see the details of that slide, but don't worry, because my point is quite simple. This is showing the different categories of clinical trials as a target. The center of the target is market-approved gene therapies in the middle. Around that are the emanating circles of the different phases of clinical trials. So at the rim is the phase 1 or phase 1-2, first in human studies, passing through pivotal phase 2 and phase 2-3 studies, filings for investigational new drugs, and then market approval is the normal process of any medicinal product. Divided into segments within this target are the different clinical specialties, which are named in orange and red around the edge. And again, amplifying what I showed in the earlier pie chart is the fact that all of the clinical specialties are represented. If you can squint your eyes, you'll see hematology, dermatological, oncological, ophthalmological, neurological specialties. And you can also see, even without needing to see the details, that around the edge, there is a large density of phase 1 studies beginning. And although we know for certain that the majority of those will not ultimately lead to market approval for many different reasons, the fact of the matter is that this ecosystem is very full, and therefore we can be confident that the predictions that in 5 years' time or less, we will have an increasing number of approved cell and gene therapies, I think resonates with many of us. Recently, we took the opportunity to look at what facilities and infrastructure was available within Australia and New Zealand in order to deliver these highly technical cell and gene-based therapeutics. And I think Australia could be suitably proud that within each of the capital cities and beyond, we have centres of excellence, including my centre in the Department of Cell and Molecular Therapies at Royal Prince Alfred Hospital, which are capable of manufacturing cells and genes for human use in a contained and safe regulated environment, which is called good manufacturing practice. This means that we seek to maintain compliance with all of the gene technology regulators' requirements, as well as those of the Therapeutic Goods Administration and any other regulatory administrations that require our compliance. So that's where I'll move on from the snapshot of global approved therapies in the infrastructure to give just now a few examples where I have had a special interest, and my group at Royal Prince Alfred Hospital and at the Centenary Institute at the University of Sydney have been involved in the development of gene-based therapeutics. So here are a few clinical snapshots and vignettes. The first one that I've been most passionate about for over 2 decades is the disease called haemophilia. The reason why that gives me such extraordinary encouragement is because I believe it to be the first ever human genetic disease identified. Back when the rabbinic scholars wrote the Talmud, the commentary on the Bible, they kept extraordinary records of families. You all know, even the non-biblical scholars amongst you, that so on begat so on, and he begat her, and she begat him, and so on and so forth. There was a lot of begatting going on in the biblical scholarly works. But basically, they were creating genealogies. And those genealogies enabled them to make observations about inheritance, even though they had no idea that there was genetic material that would convey that or the mechanisms by which it could be conveyed. They were able to observe phenotypes. And the holy covenant of the Jewish people with their God was ritual circumcision. Moses had received instructions to advise him that all male offspring were to be circumcised at birth. And that holy covenant with God meant that all male children received circumcision, and it was observed during that process that some babies sadly bled to death at that process. I pause for a moment at that striking observation and record because it is one that we should observe with some level of moment. On occasion, mothers suffered the double tragedy of their second son dying at the hands of the mohel who undertook ritual circumcision. The Talmudic scholars commented on that, and they observed that if a third son passed away from ritual circumcision, the mother was absolved of the holy covenant with God to circumcise their third and succeeding son. It was a bit ambiguous whether it was the third or succeeding. Personally, I put that under the heading of common sense. It seems to me that it's a reasonable thing that anyone would say if it's been very, very bad 3 times in a row, don't keep doing it. But there was more to that than meets the eye, because the scholars had their genealogies and they made further observations. And they said something more than 2,000 years ago that still sends shivers down my spine, because they said that not only was the mother absolved from that responsibility, but her sisters were absolved as well. At that time, the Talmudic scholars understood that there was something inherited along the maternal lineage that was reflected by a disorder in male offspring and male offspring alone. Probably the first demonstration of haemophilia and of any genetic disease that we know of. Haemophilia B is the less common of the 2 types of haemophilia A. It's caused by a deficiency in a blood clotting factor called factor IX. It affects about 80,000 people worldwide, and it causes spontaneous bleeding into joints, soft tissues, and muscles, often associated with trauma, but can occur spontaneously, in particular in the joints, the elbows, the knees, the ankles, for example. And it can be life-threatening if a bleed occurs in the skull because there's nowhere for the blood to expand into. Therefore, people with haemophilia, in a sense, live with a ticking time bomb all the time, not knowing whether they're going to have a knock on the head or a bleed into their skull, which might ultimately be lethal. Fortunately, for many decades now, for over 50 years, we've been able to purify or make available the concentrated factor that is required to reverse that clotting predisposition. And so, either recombinantly or plasma-derived, we are able to provide sufficient factor for people in Australia to be able to prevent their bleeds. We also now, by virtue of recombinant technologies, are able to avoid the tragedy that befell many people with haemophilia, namely HIV, AIDS, as well as the hepatitis viruses. Nonetheless, we believe that 2/3 of individuals worldwide do not receive any form of therapy for their haemophilia, and they suffer horrible, painful bleeds in their joints with nothing more than mild analgesia. There is only one cure for haemophilia known, and that is a liver transplant, because the source of these clotting factors is in all of our livers. Aside from that very serious procedure, there is no known cure, and therefore patients need to receive lifelong factor concentrates that are injected every few days, and some of the long-acting ones now every week or so. Therefore, haemophilia is a strong candidate for gene therapy because the clinical manifestations are attributable to a single protein. And I want to emphasize that now, that the current technology that we've harnessed to perform this magic trick of introducing new genetic material is best targeted to so-called single gene disorders like haemophilia, rather than diseases which we recognize have multifactorial or multiple genes involved, such as, for example, various types of cancer, degenerative brain diseases such as Alzheimer's disease, and diabetes. So the goals in this bleeding disorder would seem to be relatively straightforward. We want to avoid recurrent injections and single episodic injections, as shown by that sawtooth at the bottom here, avoid the risks of blood products, and obviously try and maintain continuous clotting factors in the circulation. As you can see at the bottom of this graph, people with severe haemophilia B in the red have less than 1% of the normal levels that I hope everyone on this call has. We know from many years of experience that if we're able to raise that level above about 12%, then spontaneous bleeding is no longer occurring. And of course, ultimately, we want to maintain levels in the green rather than the episodic sawtooth levels, which of course at their trough may lead to a predisposition to bleeding. The means by which we ferry the new genetic material into cells is what we define as using vectors. These vectors are typically viral, and the safest viral vector that we have harnessed to date is called adeno-associated virus. It is an AAV vector known to lack pathogenicity in humans, but infects human cells and individuals. Indeed, about half of us are exposed to this virus in the wild in the first few years of life. We genetically modify the AAV to make it unable to replicate, unable to copy itself. It's able to deliver the gene, but then it stops in that target cell. It's predominantly non-integrating, and this therefore avoids one of the major potential toxicities, because I want to emphasize at the current level of technology, we are not capable of genetically editing the specific abnormality. What all of the clinical trials have done so far have been to introduce genetic material semi-randomly into the genome and then thereby adding the gene anywhere into the genome. And therefore, the AAV system, by not injecting its payload into the actual chromosomes, but maintaining it so-called episomally, means that that is a safety factor. Nonetheless, if the gene gets into a long-lived cell, such as a stem cell or a dormant cell, it can establish lifelong correction. And we have multiple different types of these adeno-associated viruses that are capable of targeting different tissues, in particular, the liver now. Can you believe back almost 20 years, we started a clinical trial in collaboration with the Children's Hospital of Philadelphia to inject the adeno-associated virus carrying the payload of the haemophilia B gene, or factor IX, into the hepatic artery, the liver artery, as shown in this X-ray on the right. And this is the first Australian patient who ever received gene therapy for haemophilia. You can see here that the study involved different doses, and it was published in articles in 2006 and 2007. And the results are shown here from the original slide. This has become a very, very famous slide in the history of haemophilia, because many experts even treating haemophilia know this slide as revealing subject E. Subject E was my patient, and he told an extraordinary story, and we learned very much from him. What you can see here on the red line is his factor levels, and the scale is 0 to 15. I remind you that all people with severe haemophilia start at less than 1%. And so you can see that within 2 weeks of injecting a single viral payload carrying the factor IX gene, he looked like he was already manufacturing large amounts of factor IX, sufficient to get him above the magic 10 to 12%. We were just about to open the champagne corks within even 2 weeks when we saw something that shocked us and caused us then to work for the next 10 years to discover what was going on. He experienced not only a drop in that red line in his factor levels over the next 2 or 3 months, but a simultaneous elevation in his liver enzymes, as shown by the two curves, the blue and the green curves. That told us there was an inflammatory process going on in the target tissue that we sought to deliver the gene to, namely the liver. And many years of research and painstaking understanding led us to then create a model which suggests that the immune system of the body is targeting the coat protein of the viral vector that we are using to deliver the gene into the target. There is an immunological response targeting the liver cells that have been genetically modified by the viral vector. Nonetheless, Fast forward a decade, and we have now reached a point where there is very, very good news. And I'll just play a little video that is is a little old, but tells the story that went on in the last decade between 2006 and 2016. It's a very important day. It's converged 15 plus years of experimental work in the laboratory and preparation for creating a gene therapeutic, hopefully, that will cure haemophilia. As a consequence of the gene therapy that's being administered right now, we hope that in future, patients will not have to inject themselves sometimes several times a week with factor concentrate that is either obtained from blood donors or from the laboratory where it's manufactured. And more importantly, internationally, the majority of people outside of Australia and in first world countries just thinking a single injection may cure a lifelong predisposition to bleeding. For many years, haemophilia has been regarded as a proof of principle. We think that it's a perfect model system to demonstrate whether we can actually insert a new gene into a person's body and have their body then start making the factor that they lack. Having demonstrated that successfully, we not only hope to be able to provide that for other people who would otherwise not have access, but also we hope that it will extend to many other genetic diseases.
We pull this out of the freezer, and the team here really has detailed instructions as to how to pull together 4 different components and freshly prepare them into an IV bag that will be used for administration. Timing's important, choreography is important. We have to coordinate closely with the clinical team to ensure that the material is delivered, at the right time and a good time frame.
Could be fair life-changing for me, like with the treatment and that sort of stuff.
And so how's the trial gone for you?
Fantastic. Absolutely sensational. They go, I haven't had any treatments or anything, any of that sort of stuff, and I've had no bleeds in any joints. And I lost both of my brothers, one of them quite early on, and then the other one, he was passed away when he was 16.
From haemophilia? Yeah,
had a brain aneurysm and which they couldn't stop the bleeding. I'd like to say thank you for the opportunity, actually.
Fantastic.
Well, that's it. Love it,
mate. Fantastic.
No worries.
So that was Mark Lee, who allowed his name to be used and who appeared with Fran Kelly on Breakfast Radio when we announced the findings of the study. And this is, to keep me honest, these are the actual clinical results that we observed in the report. This is a graph of the months following vector administration, and I would emphasize it's just a single injection, no longer in a vessel, in a cannula that's supplying the liver, but now just in a peripheral vein. So it's a simple intravenous infusion over less than an hour. On the y-axis, you can see the factor levels now with a scale up to 100. If you have severe haemophilia, as I've mentioned, you're less than 1%. So that's the little yellow dots down the bottom. And these are the first 10 patients who were treated with a single injection of this viral vector called adeno-associated virus carrying the factor 9 gene. All of the patients received a therapeutic benefit of the first 10, and the best of all was the one at the very top there in red. That was Mark Lee. Here's the data. Prior to gene therapy and after gene therapy, the number of annualized bleeds per annum, the bleeds per annum shown on the left, and after gene therapy shown on the right, and that data is published, and I won't belabor it, but we now have 5 years follow-up on these individuals, and indeed 15 years of data safety from the first patients that I showed you injected in 2001. There are still hurdles to overcome, and they are twofold. It's still the immune system giving us trouble, both humoral immunity, the antibodies attacking the virus when it's injected, and then the cellular immunity, potentially a memory response that is attacking the liver cells once they're genetically modified. And we need sometimes to administer steroids to treat individuals who are having an immune response. And this is Mark himself, who required steroids to suppress the immune response, but then after that had an outstanding response and has now got almost completely normal levels of factor IX in his blood. Many years after the treatment, he's around 90%. And there he is smiling in that shot that you've seen before. So in summary, haemophilia gene therapy is looking very good. It is not yet approved, but there are multiple companies worldwide that are sponsoring clinical trials in phase 1, 2, and 3 of clinical development that are looking very, very promising. And indeed, in Australia, CSL has just purchased a company called Unicure that has advanced gene therapy development for haemophilia B and indeed for haemophilia A. Let me change tack as time is getting on to another disease, because I mentioned haemophilia as an in vivo gene therapy. Thalassemia is also now a blood disease which is much, much more common than haemophilia. Thalassemia, would you believe, is the most common inherited blood condition in the world. It is an inherited abnormality of the hemoglobin gene. We as humans have co-evolved with the malaria parasite, and people with mild thalassemia or thalassemia intermediate even have a semi-resistance to the malarial parasite. Nonetheless, in the severe form of transfusion-dependent thalassemia, these individuals require lifelong blood transfusions, which leads to iron overload and toxicities from that which can be life-threatening. The idea of gene therapy is to take blood stem cells out of the body from these individuals suffering from this inherited abnormality, genetically modify them outside of the body with a viral vector, in this case called a lentiviral vector, and then return those cells to the individual after they have received a form of chemotherapy that, in a sense, makes space for the now genetically modified cells to return to their home. We were involved in 2 clinical studies called North Star 1 and North Star 2, which involved the different subtypes of haemophilia. And again, these data are published in the last couple of years, and I won't belabor them, but they essentially demonstrate that it is possible now to achieve a therapeutic benefit in patients with so-called beta thalassemia. I want to show you the design of the study of this ex vivo therapy because it highlights important aspects. We are able to mobilize the blood stem cells that normally reside in the bone marrow into the peripheral blood and then siphon them off using a technology called leukapheresis, shown in the top left there. The cells are then purified or removed from the body and then shipped to a centralized manufacturing facility where they can be genetically modified with the virus that I just mentioned and then stored frozen indefinitely. At the same time, when those cells are then checked for safety and that they have no problems with, for example, bacterial contamination, they're sterile, they don't have any other issues, we then treat patients with so-called conditioning regimen of high-dose chemotherapy. That is required, and that is the main reason for any toxicity in this study. The cells are then, within a couple of days, infused back into their own body, but now, of course, they are genetically modified, so they are capable of making the gene that those patients originally lacked. And of course, then we do exhaustive follow-up. The results of the study have been extremely positive, and I won't belabor them other than to highlight the fact that now thalassemia appears to be curable using this form of gene therapy, the evidence being not only a publication in the New England Journal, but the fact that the European Union has approved for specific patients provisionally to be treated using this technology. The cost is jaw-dropping, and I don't have time to talk about the costs of these technologies, but I would be happy to do so during question time. But as you can see there, $1.8 million over 5 years, but of course they give you a little insurance policy, which is if you perchance do not respond within the first year, you don't have to pay for the rest of it. Nonetheless, this technology is changing lives, and at Royal Prince Alfred Hospital, we were very proud to treat some of the earliest patients And this is Jack, who also allowed his name and his image to be used. And I'm very proud of this image because this was a photo I took of him in the clinic where he was receiving the very last blood transfusion that he ever needed to receive after his 35 years of life, where he received blood transfusions every month. This was the last one he received before his bone marrow transplant of his own genetically modified cells. And since that time, more than 3 years ago, he's never had to receive another blood transfusion ever since. And he's very pleased about that with his wife and 3 children. The last quick example that I'll give is CAR T-cell manufacture. And that's in a sense even broader because that's targeting cancer. CAR T-cells are now approved in Australia, at least for leukemia, using a technology developed by a company called Novartis in collaboration with the University of Philadelphia, and in particular a group led by Carl June. The idea here is to genetically modify one's own immune cells so that they're now capable of recognizing cancers in one's own body. In a sense, it's reprogramming one's own sleepy immune system to target cancer. The response rates in lymphoid leukemia are nothing short of extraordinary. And indeed, if you don't believe an 85% response rate in patients who have no other option and have received all of the other previous treatments and are looked to be at death's door, believe the market. The company that was developing one of these 2 technologies, called Kite, was sold for US $12 billion before they'd ever sold a single drug, and they were purchased by Gilead a few years ago now. These cells are manufactured by taking immune cells out of the body using a technology similar to that which I mentioned before, siphoning cells off the body, but in this case, specific immune cells. The cells are then shipped to a centralized manufacturing facility where they're genetically modified outside of the body. They are then stored and checked for safety and then returned to the individual for injection, now having been reprogrammed. The mechanisms by which they are acting is basically the reprogramming gives them the ability to now recognize and kill target cells with specific markers on them. And in the case of acute lymphoblastic leukemia and other malignancies of the blood cells, this is a marker called CD19, which I won't talk about. Nonetheless, there were 2 clinical trials that we were involved in, pivotal clinical trials, that led to the approval of these drugs in Australia. And here are these cells in a scanning electron micrograph wrapping themselves around beads which are coated with CD19, showing not only the exquisite specificity for these cells, but also their exquisite beauty and the way that they wrap themselves around these smaller beads. This is again emphasizing the fact it is a living therapy. Not only is each one of these single cells capable of killing thousands of tumor cells, but they are also capable of proliferating. And proliferate they did in one of the very first recipients of this treatment, Emily Whitehead, who at the age of about 7 had, without any doubt, only a few days or indeed a week to live after all of the different therapies in her acute lymphoblastic leukemia had failed. And she's now, in fact, 7 years post a single injection of these reprogrammed CAR T cells, which has led to her apparent cure and obviously a life that is now enjoyed with her family, and she has established a foundation, which is one of the biggest foundations in the world, supporting the use of CAR T cells. Again, this emphasizes the fact that these CAR T cells are targeted to the specific marker I mentioned and a living therapy. Once they've done their job, the cells remain in the body for a long period of time, monitoring to see if the leukemia returns. But if it doesn't return, the cells are there dormant and indeed may ultimately disappear. This has now become a 5th pillar of cancer therapy, added to the familiar pillars: surgery, chemotherapy, radiation, and other targeted biologicals. And indeed, our Health Minister, Minister Hunt, has approved those therapies in Australia for acute lymphoblastic leukemia and lymphoma. In Royal Prince Alfred Hospital, with senior colleagues, including Nico van Zandewijk and others, we are developing mesothelin CAR T-cell therapy for pancreatic cancer and other solid tumours, which is the next big challenge to develop this technology to affect other cancers beyond blood cancers. And so these CAR T-cell therapies are being developed in our facility against pancreatic cancer, which has still a woeful outcome. As many of you would know, pancreatic cancer has a very, very poor prognosis. And even over the 20 plus years that we've been monitoring 5-year relative survival rates, you can see the graph has barely even touched 10%. We have published on this in a lovely review by one of our fellows and PhD students, Gerard Chu, where we have characterized the immune microenvironment that prevents the success of these CAR T cells in other tumors. And indeed, Daniel Yeo in our lab is studying the circulating tumor cells that are in the Southern Hemisphere technology based on RareSite to identify tumor cells and see how well we're doing. We've sought funding and been successful in receiving that funding from none other than the Li Ka-shing Foundation from Hong Kong. And this is our friend Tony Biddle on our consumer advisory panel, who's very enthusiastic about that because he is a long-term survivor from malignant pleural mesothelioma. I'm mindful of the time and have a few minutes to go, but I'm also mindful that all good speakers realize when they've got 20 minutes of question times, if they bump into the question time, they can steal a little bit of that. Susan, if you'll just indulge me a couple more minutes, please. I've just got a little bit more to say on one more study and then just touch my cap to the responsibilities of clinical innovation. So the last example that I will give is pluripotent stem cells, because I'm very excited to share with you some data that is only about to be published in the next few weeks. And this is in a very devastating and life-threatening disease called acute graft versus host disease that occurs in patients who have received a bone marrow transplant from someone else, so-called allogeneic bone marrow transplant. This is a greatly feared disease with a high mortality if it is not treated early on with steroids, and indeed even the majority of those patients may well have significant morbidity affecting their liver, their gut, or indeed their skin, as shown in the images. Mortality rates can be as high as 90%, and there is no approved therapy available in the world for this devastating disease. Many of you would know that Sir John Gurdon and Shinya Yamanaka back in 2012 received the Nobel Prize for Medicine and Physiology for their discovery of induced pluripotency. That is a type of stem cell that we reviewed many years ago that basically is like embryonic stem cells, but overcomes some of the ethical challenges that embryonic stem cells have been burdened with. Using that technology based on an invention from Jamie Thompson and Igor Sluvkin, we've worked with a company called Cynata in Melbourne to produce mesenchymal stromal cells, which overcome many of the challenges that would otherwise beset the use of induced pluripotent stem cells. And indeed, those cells have been stored and are made available in our Cell and Molecular Therapies department, a laboratory complex which I show here, and which is represented in these images. It's like a satellite, like a spaceship where the air is more than 1,000-fold more pure than the air in the normal air that we're breathing right now. And the clinical results of the study shows that the majority of patients, indeed over 80%, received a very good response. This clinical data is very reassuring and mirrors data generated by other studies, but I want to emphasize that in this study, we have achieved what has not been previously achieved, namely the first completed human clinical trial of induced pluripotent stem cell-derived cells in any disease, and a very encouraging result, which of course requires further safety studies and a much larger cohort than the original 15 patients that we treated. So in the last minute, I just want to touch on some of the responsibilities of clinical innovation, which has otherwise been referred to as the dark side of cell and gene therapies, because it's been an area that I've been passionate about and have published and spoken widely about over many, many years. Because if you believe that these technologies now offer hope and promise to patients with unmet clinical needs, we have to also recognize the fact that there are many other areas of human endeavor which converge perhaps on science in the center, but that have incredibly important components that need to be considered and will contribute substantially to the development of these molecular and precision medicines. Unfortunately, there are many people in our community who would take advantages of some of the false claims that have been made by individuals, and we've had the opportunity to publish examples of those in The Guardian over several years with my colleague Carl Power, We've had the opportunity to reveal direct-to-consumer marketing worldwide, and the fact that Australia is well represented in shonky operators and cowboys and cowgirls offering unproven cell-based therapeutics to individuals for any disease that ails you. We've published reports showing that some unproven cell-based therapies may be futile, in particular those derived from adipose tissue, that they've been marketed directly to individuals, and that they have really provided only false hope to individuals who deserve nothing more than true hope and real therapies. Indeed, Carl Power and myself have a book coming out early next year called Flesh Made New from HarperCollins that will elaborate on some of those false claims and where the fraudulent operators have moved. If you want to hear more about that, you can listen to the Boyle lectures, which are still available online, four 30-minute lectures that I was privileged to deliver in 2018, invited by the ABC Board of Directors. So in conclusion, we have to ask ourselves at this pivotal dawn of a molecular medicine, what level of evidence is required for a procedure to become standard of care? On the left-hand side, there are proven-based therapies such as bone marrow transplantation, corneal replacement, some skin-based therapies. On the right-hand side, there are potentially emerging therapies, but that remain as yet unproven. And if individuals like us are confused about whether or not they have reached the standard of care, just think what it's like to be a member of the community who can't distinguish that based on the empirical evidence. It is that grey zone that shonky individuals take advantage of and separate out the proven from the unproven and blur the separation. We as a group of scientists and individuals committed to empiricism, I think, need to commit always to empirical and evidence-based medical data as well as other data, rather than a market-driven proposal which some would support. I believe that one day many drugs may be replaced by cells and gene-based therapeutics. I want to acknowledge the fact that I have a large team behind me, some of whom I've mentioned during this presentation and some of whom have joined us online, the Department of Cell and Molecular Therapies, Royal Prince Alfred Hospital, and the Gene and Stem Cell Therapy Program at the University of Sydney Centenary Institute. I acknowledge our funding bodies, and I would very much welcome questions. Thank you very much.
John, thank you very much. That was a fabulous yet very grounded presentation. And I listened to your Boyer lectures, and you've improved over and above those, which is a very hard task to achieve. I'd like to turn to a quote as the first question that you made or said at the Boyer lectures, and get you to unpack it a little bit for us. You said, "We stand at the start of a revolution that may alter the very fabric of our being and the essence of what it means to be human. Reproductive technologies and gene and cell-based technologies offer the possibility of prolonging life and curing disease, even controlling our evolution as a species." Would you like to spend a few minutes unpacking that for us?
Perhaps I was being too profound. Thank you for the invitation, Susan. I chose those words very carefully, and I recognise that they do need unpacking. They're made in the context of a legal reality which is currently under review, namely that germline gene intervention is illegal in Australia and is prohibited in almost all jurisdictions worldwide, either by law or by practice, in particular in the United States of America and the EU. Many of you will have heard last year of Shiwan He, who in Shanghai produced so-called CRISPR twins or CRISPR babies. That was indeed an attempt to genetically modify human babies with an intervention that meant that they may or may not be resistant to the human immunodeficiency virus. This scientist who was trained partly in the United States of America is now in jail for his infractions in China to universal condemnation worldwide. In other words, a pushback by the world's scientific community that it was not allowed to overcome the barrier of genetically modifying that which is inherited for many, many reasons. One of which, of course, is that there is a problematic issue of consent. You can't get consent from someone who's not born or even conceived yet, and that's just the beginning. In this particular egregious example from China, the infraction was even worse because there was no disease being treated. It was an attempt at some form of prophylaxis that was obscure and ill-founded. Nonetheless, Australia is currently grappling with a real alternative option, and that is of mitochondrial disease therapies. You would know that there are some mothers who bear babies who suffer from a disease or a series of diseases, more than 30 different subtypes that affect the mitochondria, the little batteries in all of our cells. And they are severe and life-threatening disorders. Some of these babies don't survive more than their first few years. Some have neurological deficits, hearing deficits, and so on. It is now possible to create what we call three-parent babies. And that means the mitochondria are, in other words, donated from a third party, a second mother, if you will. But like it or not, that means that those babies born would carry the genetic material of three human beings. The mother and the father in the nucleus, and the second mother, if you will, in the mitochondria. It is currently illegal to undertake that, and I was part of the NH&MRC advisory panel that provided advice after the Senate committee did earlier this year indeed, and preceded the Senate committee. We will need legislative change to allow three-parent babies in Australia. The UK has just begun it in the last couple of years, and indeed, if that is allowed, it will potentially open the possibility of germline gene intervention. And I'll simply close, Susan, in the answer by giving some examples that I've just done by saying the following. When we provide informed consent to individuals like Mark Lee with haemophilia, we insist on them understanding that when we give them their gene therapy, hopefully to cure them, we are absolutely honour bound and legally bound to prevent any germline gene modification. We are prevented, and we must not overcome any germline genetic barriers. So the patient's offspring, of course, will suffer from haemophilia or be carriers of haemophilia. Patients always ask us, why don't you just cure my whole genetic material once and for all so that I no longer carry the burden of this disease that I never wanted, I didn't do anything to get, and I don't want my kids to get it or carry it. And so the very question that many of them ask is one that I think carries enormous ethical weight, and it's one that we need to consider as we go forward, because we are at the dawn of this new medicine and we have harnessed the technology. It is now an ethical, moral and legal question as to whether or not we embark tentatively towards crossing this germline.
Thank you very much. Now, that does answer the question that has come in from several people, including Judith Wheeldon. Another question, how do you see the dual use problem with these technologies? For example, could they be weaponized?
Yes. There's a lot spoken about dual use, and of course we consider it in the Office of the Gene Technology Regulators discussions from time to time when this does come up. Clearly, the weaponization of genetically modified organisms is abhorrent and not something that anyone ethically inclined or indeed a regulator would countenance under any circumstances. Nonetheless, I'm reminded of the dual use of the kind of technologies we're talking about when they pertain to genetically modified crops vis-a-vis genetically modified humans. Many of my patients like to refer to themselves as GMOs or genetically modified organisms. And of course, the legislation excludes that definition. Human beings are excluded from the Act, the Gene Technology Act of 2000 in Australia, because of that very fact. But the point is that the same gene technology that we've harnessed to create frankenfoods, if you will, has a dual use, which is to cure human diseases. And it is surprisingly similar, and the concepts are surprisingly resonant. Nonetheless, of course, there are differences between plant genetic technology and mammalian genetic technology. But it serves to emphasize that I think there is a misunderstanding in the public's view about what is safe and what isn't safe. Because I can assure everyone on this call that the level of rigor that we pay and attention that we pay to making sure that crops, for example, are safe, and when approved for an intentional release, they are safe for all humans and to the environment. This is something that is a requirement of the legislation. And yet that technology is similar to the technology used for human GMOs. I can't exclude the use of this technology for gene doping in sports, which of course we don't countenance. I can't exclude its use in weaponizing so-called other viruses and so on. But truthfully, that technology is already widely available and widely acknowledged. It could be used anywhere. It could be used by rogue states. There's no stopping that other than an international consortium of like-minded individuals and governments exercising influence and condemning any such activities.
Okay, two more questions, if I may. The first one is the use you raised towards the end of your presentation on of CAR T cells in solid tumours, pancreatic cancer, for example, where access of drugs or any treatment to the cancer is difficult because the tumour can be impenetrable. What are the challenges, particular challenges in these solid tumours to be overcome by CAR T therapy?
Yeah, it's very much the holy grail of CAR T cell technologies, and I did make mention of some of the challenges and a couple of the references. But in short, we all ask ourselves, why is it the case that CAR T cells have been so successful against CD19 leukemia? They're not quite as successful for the solid B-cell malignancies that carry the same marker, CD19, but there is a difference. You've painted an image in everyone's mind, Susan, which is very resonant, namely these dense tumours that the immune cells can't get into. Well, it's certainly the case that they do have a fibrous tissue sometimes intervening in them. But what we, I think, have come to understand that it is much, much more complicated than that on a molecular basis. And there are many ways that cancers evolve to, in a sense, turn themselves into a cold or invisible cancer. And that is to say that they downregulate markers, or they indeed upregulate specific suppressors of the immune response in order, in a sense, to protect themselves. In a sense, like a Darwinian evolution, the cancers evolve in the body to protect themselves against an immune response and therefore survive. Whether that's an inappropriate image or not, it seems to be the case that that's what's occurring. Most CAR T cell technologies as they currently stand have not been able to overcome this so-called cold immune environment. One of the ways that we've sought to overcome it in collaboration with our academic colleagues at the Memorial Sloan Kettering Cancer Center in New York is to use an armoured CAR T, so-called. Armoured because it is carrying not just the CAR, the chimeric antigen receptor, which is targeting these particular T cells, these immune cells, to the marker called mesothelin that's present on pancreatic cancers and mesotheliomas. But in addition, we have another gene that those T cells express called PD-1. And PD-1 is a suppressor of the suppressors of the immune response, in a sense. And it's like a soak, or in a sense, a decoy that armors the T cells so that it is no longer suppressed. And we believe, and there's good animal data to suggest that when we do put these into patients, they may overcome some of those barriers. And last but not least, we have a highly innovative delivery system wherein we plan to deliver these chimeric antigen receptor armored CAR T cells directly into the tumours using endoscopically directed injection via the gullet, the stomach, and then straight through into the pancreas through a needle, thereby overcoming the homing challenges of those cells. So combining those approaches, we think we have a chance to be able to really overcome this major hurdle that has remained problematical over the last few years to attack solid tumours with bona fide markers.
Okay, and the last question from Nairi Palmer. I think you should give us a score from on a scale of 1 to 10 about your optimism for commercialisation in Australia of some of the technologies you've discussed.
Yes, I think we have been a little behind in our embrace of commercialization of those technologies. I'm very, very proud of the fact that we have an extraordinary environment in Australia that has amongst the best basic and translational clinical scientists in the world. We secure many of the clinical trials that are pivotal, and I've given you examples of some of the world's first clinical studies that have been completed in Australia. We have infrastructure that I've shown during my lecture, and we also have a very advanced hospital system, which is capable of giving the highest quality care to individuals who are citizens. And of course, everyone is proud of the fact that that is free for anybody in Australia who is a citizen. Nonetheless, capturing early phase intellectual property, which ultimately is the basis for commercialization, has been something where I think we've been somewhat derelict and somewhat behind. And I reflect on many aspects as to why that may or may not be the case. It perhaps has to do with our risk, our desire to embrace risk or fear of risk. It also has to do with capital, and indeed it has to do with the availability of managers who understand the very complex process of taking an academic invention all the way through translational evidence into the very, very expensive phase 1, 2, 3 studies. And often individuals need to be mindful that they're not capable of spanning that entire gamut from early phase academic discovery all the way to clinical commercialization. Nonetheless, there are emerging companies in Australia which have a track record in achieving that. CSL is one of our best, ResMed is another, Cochlear is another, and then there are smaller companies. Mesoblast is no slouch in that, and it's just recently had an approval in the United States of America. Sinatra I mentioned, and there are a number of other countries. But we can go, there are many other companies, but we can go so much further by providing tax incentives, by taking advantage of pots of money that people like Glenn Begley and BioCurate have available, and many of the universities are growing. With the COVID setback and the financial pressures that are going to be impacted on our universities, this is now going to be an increasing challenge. And I know that there are organisations shopping around the world looking for intellectual property to purchase from Australia. So I think it is a real and urgent problem. Australia has a true opportunity to take advantage of these great developments, but nonetheless we have to be wise, and I'm afraid we have to spend quite a bit of money in order to secure them, because they'll be taken offshore without any doubt in the coming years, and indeed there are many examples where that's already occurred.
Okay, John, I'm not going to thank you. I'm going to hand over to my fellow Vice President, Judith Wheeldon, to do the honours.
Thank you, Susan.
It is, in fact, my very great pleasure to have this opportunity to thank you on behalf of all of us. I absolutely know that's true. Professor John Rasko, AO, first of all, we thank you for the work you have done, having nothing to do with this talk, but the progress that you have forged in a most important area of science, medical science, and human life. That is a great contribution already. In your Boyer lectures, which I also went back and listened to again, I was very, very impressed with the moral and ethical issues you raised and emphasized so articulately, and I would encourage people to go and listen to those again. But specifically tonight, I felt full of joy and quite cleansed listening to you. And that's an odd thing to say after a lecture on a very sophisticated topic, and one where you didn't pull your punches. In a very brief period of time, you gave us a lot of detail, a lot of knowledge, and many clues about where we should be directing our interest next. So why did I have that feeling of joy? Excellent presentation may have had something to do with it. But really, I think it's magnified by the fact that we are living in a rather dark and difficult time. COVID, obviously. And we talk flippantly or easily or lightly about what we will be like, what we will learn from our COVID experience. And I think the benefit of tonight's lecture points us a little bit in that direction. Because the other thing that is making this a dark time is the uproar in our political lives around the world. What's going on in Hong Kong? The threat to Taiwan, what's going on with the Uyghurs, and probably more specifically and directly relevant for us, the Black Lives Matter, where we have tremendous pain and turmoil existing from our past here and in the United States. So it is a dark and worrying time. But you have spoken to us about science and what clear thinking can do, and how if we approach issues intelligently with historical knowledge, and I did love going back millennia to the rabbis, and it was most interesting to have it posited that that is the first medical science, and I'm quite happy to accept that. But it was wonderful to hear and what could be an abstruse topic, but one that is very, very important to our lives, presented in a very clear, moral, ethical, and humanitarian way, so that we could listen and accept responsibility for finding out more about this and thinking about the issues that do arise. So thank you for that presentation, and thank you for giving us the human drama of the successes over the years, the people whose lives are really affected. The reason we work so hard for the Royal Society of New South Wales is because we have the opportunity to hear lectures like this and to, in fact, meet the person, unfortunately not face to face, but maybe that's in the future. You made it feel like a genuine social occasion. You presented us with enthusiasm and joie de vivre, along with a great deal of learning. So we finally thank you for what you did. You begat a wonderful evening of social interaction and deep learning, and pointed us in the direction that we need to go next. So thank you very much, Professor John Rasko, and I hope you can hear the applause around New South Wales. Thank you.
Thank you so much.
My pleasure.